The human body has over 600 muscles, all of which that we use for daily activities ranging from our simple motor functions like walking and breathing, to managing our internal organ processes. At some point, these muscles have a limit in how much they can grow. That no matter how much you may work out these muscles, they will not get any bigger. However, what if I told you that without ever hitting the gym once, you could essentially double your muscle? Sounds impossible? Well, in the early 1990s, a geneticist at Johns Hopkins University named Si-Jin Lee found a way to do this when testing laboratory mice. How did he do this? He did this through the use of transgene and myostatin manipulation to stimulate muscle growth. Si-Jin Lee cloned genes that coded for a group of proteins known as transforming growth factor beta. These cloned genes are called growth differentiation factors. The scientists used gene editing and were able to make genes lose their functionality. They used this method, and by breeding mice that lacked only one of the 15 different genes, they were able to isolate and determine what each growth differentiation factor did. You see, mice without GDF1 were born with organs on the wrong side. However, mice without the 11th gene were born with an extra set of ribs. Having 36 ribs caused health problems among the mice, and they were unable to survive for very long. However, mice without growth differentiation factor 8 not only survived, but had double the muscle. The gene GDF8 is located on chromosome 2. Lee named this gene and its protein myostatin. In Latin, the prefix myo means muscle, and the suffix statin means halt. This name is fitting since the main functionality of myostatin is to signal muscles to stop from growing. Without it, muscle growth may explode as it did with the mice. Since then, scientists have worked to find out if this muscle growth mutation has the same effect in humans. The only recorded occurrence of this mutation in humans is when 3 years earlier, a German boy was born with excessive muscle. More research is currently being done on this topic because it is essential to combat diseases like muscular dystrophy. On December 5th, 2019, SpaceX-19 launched from NASA Kennedy Space Center on a resupply mission to the International Space Station. However, there were some unlikely hitchhikers aboard the space shuttle. A total of 40 mice were loaded onto the shuttle as part of an experiment for myostatin muscular dystrophy research. Of the 40 mice, about half the mice were treated with myostatin inhibitors prior to taking off. The mice stayed on the International Space Station for about 33 days. The mice from the experiments showed that the myostatin inhibitors were effective for muscle growth even in the setting of microgravity. The mice with myostatin inhibitors had lost less muscle when compared to the regular mice. More tests showed that bone mass density was also higher in space mice than ground mice, proving that these inhibitors gave protection against muscle loss, as well as loss in bone mass density in zero-gravity environments. After the flight, the mice returned to Earth so that the scientists could collect their data. After only 2 weeks back on Earth, the mice seemed to recover all of their original muscle. This result shows promise given that the astronauts returning from the long missions in space take a while to recover muscle and bone mass. Through further research with myostatin inhibitors, we can find a way to reduce their recovery time. As we journey further and further into space, the longer missions will have substantial effects on astronauts' bone and muscle mass. Through the use of myostatin inhibition, scientists believe that they can combat bone and muscle loss during the flight, as well as increase recovery times after the spaceflight, proving that these advancements in this area of research is vital for space exploration. Duchenne muscular dystrophy is a genetic disease that usually occurs in males. It causes muscles to be wasted, leading to heart problems and loss of skeletal muscle. This seriously life-threatening disease may require the use of a wheelchair, as the loss of muscle prevents those infected from standing or walking. In an attempt to fight this disease, Pfizer researcher Michael St. Andre conducted a study that focused on using a specific antibody as a blockade for myostatin. This antibody called MRK35. Antibodies have a chain that binds to an antigen, which in the case of MRK35 is myostatin. At the antigen binding site, MRK35 binds with the myostatin. The antibody then inhibits the transforming growth factor beta ligands from binding to cells. This prevents myostatin from signaling the cell to stop muscle production and leads to increased muscle weight and bigger muscle fiber size. Currently, this research has only been conducted on mice, rats, and primates. Further advancements in this research may allow humans suffering from muscular dystrophy and live normal lives.